my pleasure. Thank you. It is my pleasure to introduce today's speaker, Professor Tilman Gernbrodt, who came here from the East Coast, from Dartmouth College. He's a professor there, to talk to us about recent advances in yeast biotechnology. Professor Gernbrodt is not only a professor of bioengineering at Dartmouth College, but he's also a very successful bioentrepreneur. Thank you, Heidi, for helping me how to pronounce that. And he has a successful history of founding several companies. One of them was sold in 2006 to Merck, Glycoproteins, which deals with the humanization of glycosylation in yeast. In 2006, also Nature Biotechnology named Gernbrodt one of the most notable people in biotechnology in the past 10 years. And to cite one of his last papers, the technology needs to follow the problem, not the other way around. And with these words, I want to welcome you here, and we're all happy to have you speak here.
Thank you, Patricia. Thank you for having me. If there's an award to be handed out for tenacity, we're not taking no for an answer. It should go to her, because it was not me deliberately trying to ignore her, but when life gets busy, sort of things get shuffled to the bottom. But she was very polite, but incredibly consistent. If that's any prediction of career outcome, I would put my money on Patricia. So unlike many of the talks you probably have here, I'm not going to talk about one particular recent discovery. I want to give you more sense of what a career may look like in this field that you guys are all in. And the reason I say that is because I was in a very similar field that you're in, and I'll talk about that later. But throughout my career, had to pivot many different times. And when you're in that situation, it's always uncomfortable. It's always something new that you didn't necessarily anticipate. But when you see that someone just sort of does it and it works out quite well, it may give you encouragement at the time when you have to make such decisions. So it reminds me of the Dutch philosopher Søren Kierkegaard, who said, you live life forward, but you understand it backwards. So since I'm a little older than most of you, I'll give you some perspectives of how that played out in reality. And so when you think of, when you think of, where's Adam? Oh, you know what?
He tried before. Not the right part. The part's in there.
Nothing's working.
Oh, it's fine.
Let's try this and start
Yeah.
again.
Okay. Now something works.
Great. Good. Thank you. So when you look at us as a species, obviously we've come a long way. But I would analogize sort of our evolution to what happens to us at the individual level, at the micro level, if you will. I certainly see things very differently than I did when I was a graduate student or as a postdoc. And sometimes, not always, people benefit from sort of hearing how people approach their specific career choices and decisions they had to make. And I thought, again, you guys may benefit from that. So what I want to do is, first of all, sort of talk about what your life looks like right now. Most of you are living in what I would call an epicentric world, where epi refers to Eppendorf tubes. And what you do is you carry out reactions where you know the inputs. You then run reactions. You analyze the outputs. From that, you develop an understanding of what goes on, and if you're really good at it, you can sort of establish certain rules of how to control reactions. And even better, if those reactions give you something that benefits mankind in some way, that's even more rewarding. But that's what most of you do at this stage in your career, and it's an incredibly important aspect of becoming a scientist, because you know how to design experiments, how to interpret data, and all that kind of stuff. Okay? So when I was doing that as a postdoc at MIT, so again, you would have to analogize that to me just having parallel toes and just gotten out of a tree somewhere in East Africa. When I was doing that, I was interested in this particular organism, or more specifically, I was interested in the enzymatic machinery that allows this organism here. I believe it's now called Ralstonia eutropha. It's changed its name so many times, so I don't keep up with the recent nomenclature. But this bug has the ability of taking in carbohydrates and converting them into a aliphatic polyester called polyhydroxybutyrate, which I'm sure many of you, or most of you, know what that is. And so, as a postdoc at MIT, I was interested in understanding the enzyme that takes a soluble substrate, hydroxybutyl-CoA, and polymerizes it into this polymer. And so that was my tube problem. So understanding the enzymology of that particular enzyme. Without understanding the larger context, which was always described to me, well, this is going to be important stuff. It's made from a renewable resource, and more importantly, the plastic that comes out of it, nature has a tendency when it makes polymers to find a way of degrading those. So it comes from a renewable resource, and it's biodegradable. What's there not to like? And that was sort of the level of inquiry that I was indulging in. Again, mostly focusing on what does this enzyme do and so forth. Okay? So again, the simplistic view was articulated by others of why this is important, why this area in general deserves human inquiry is this poster here. It reads, "It's the mother most diaper companies totally ignore." And what it says is really nothing. It just is, the net product of this is supposed to be guilt, right? You're supposed to feel bad if you use plastic or plastic diapers. And when you do that, there's, of course, an arbitrage opportunity between your guilt, their bank account, and your wallet, right? So that's the main product of this. But it doesn't really inform or tell you anything about why plastics are bad, other than sort of evoking this feeling. But most of you know the main criticism against polymers made from fossil carbon are that we're making them from resources that are non-renewable. You know all this. I'm not going to bore you with that. This typically shown in dreary black and white, ugly colors, and this is the future where everything's going to be nicer and greener. So that's one thing. The resource is renewable. And the second thing is that we all know that plastic bags degrade very, very slowly, if at all. And so there's an environmental impact by making polymers that don't degrade in nature, and they accumulate in places where they're not pretty to look at. So that's problem number 2. And all this can be solved if you could make plastics from a renewable resource that both addresses the issue of fossil carbon depletion, supposedly, and secondly, when you make materials out of PHB and you put them into an active compost environment, there are microbes in the ground that secrete enzymes that break down this polymer and ultimately lead to its degradation. Okay? So the good news is there are plastics that have thermoplastic properties, like similar to perhaps polystyrene, that we can make from a renewable resource and they degrade. So that's the good news. And that is typically illustrated in illustrations like this that show the life cycle of the sun, leads to the sequestration of carbon. You all know this. That gives us sugar in the process that we call corn wet milling. We grow about 75 million acres of this stuff to give us sugar that we currently use mostly to make ethanol and fuel the obesity and diabetes epidemic. But we could use some of it to put into microorganisms that then make this plastic, and this plastic upon disposal degrades into CO2, so what's there not to like? Okay? So I worked in this area first 2 years as a postdoc at MIT, then joined a biotech startup company. This was long before the current sort of bioenergy craze. This was in the early 1990s. Basically to commit myself to helping make this process work and be more efficient and all that kind of stuff. It's not until literally 7 years later that I asked the question, if we're displacing one technology with another technology, what are the actual trade-offs? What are the real trade-offs that we have to be concerned about? And ended up asking a question that now seems pretty obvious, but at the time was not so obvious, believe it or not. Namely, how green are these green plastics that as we substitute one for another, what are the specific trade-offs? And so myself and a colleague of mine that also had worked in this area for many years, Steve Slater, conducted a few life cycle analyses and came to a rather surprising conclusion. So what you see on the left here is, what does it take to make polystyrene? And the take-home message really is, you need about 2.3 kilograms of fossil carbon inputs, in this case mostly natural gas and oil. That's your input. And a big chunk of that input actually is combusted in the process to give you the energy to do the distillations and all this stuff, to give you 1 kilogram of polystyrene, and then you can make plastic cups out of it, or like these clamshells to, carry your salad around, that kind of stuff. So it's heavily used in the food packaging industry. So we said, okay, fine. If that's what it takes to make plastic from from fossil carbon, what would it take to make plastic from basically a corn-based process using this technology I described before? The first thing you have to ask yourself is, what happens when you do corn farming? It turns out corn farming is by no means a fossil carbon neutral activity. In fact, the USDA very rigorously monitors and understands how much fossil fuel equivalents go into corn farming. Most people think it's tractors driving around. It isn't. Most of it really is industrial fertilizers. But we can calculate how much does it take to make 5 kilograms of corn, which ultimately lead to 1 kilogram of PHB on the back end. So when you do that, you get 3 kilograms of starch. You get 3.3 kilograms of glucose after you hydrolyze that starch. And so we can calculate how much fossil fuel inputs we have from statistical data. You don't have to do anything, just look at the data that the USDA provides. And of course, this differs geographically. There are states where you need irrigation, so there's a little more, but we roughly know what this takes. The next thing is a fermentation process, and any process engineer can look at a process. You need agitation, aeration, all that kind of stuff, cooling when you get into higher cell densities. And again, you can assess very accurately what it would take to run such a process at a commercial scale, where you convert 3.3 kilograms of glucose into a kilogram of PHA. Okay? So then you have a bug that contains plastic. That's not something you can injection mold or do anything usefully, so you have to get out of the bug, wash it, clean it, and there's some expensive energy by centrifuging and washing and breaking the cells open to get it out, to ultimately end up with a kilogram of PHB. All right? So now you have the 3 steps. You add up the energy that is required to do that, and what we found, to our surprise, is you need about 2.4 kilograms of fossil carbon to run the whole thing. So while the carbon that you're moving from the field into the plastic that you're consuming indeed is renewable, but the energy you need to make all this happen is not currently certainly not renewable, and so therefore the amount of fossil fuel that you're expending to make this is actually significantly higher than over here. More importantly, this is all process energy, and as we know, process energy derived from fossil carbon leads to CO2 emissions. So this is all burned here, just like this is all burned here, but as you know, this number, or as you can see, this number is almost twice the amount that is over here. All right? So if you want to make large-scale plastic from this process here, you have to be prepared to emit more carbon into the atmosphere. It just is what it is. Okay? And as I said before, the net CO2 contributions are not particularly favorable. So in summary, this is all true, but what makes the story more complicated is it is omitting certain material or energy streams, which are growing corn doesn't happen without fossil fuel inputs. Converting corn to sugar doesn't happen without fossil fuel equivalents. So you can see that as you attribute, or not attribute, as you account for all these energy and material flows, the picture is a little less encouraging. And the next thing is that this thing about biodegradation, most food waste ends up in landfills or composting facilities, which means it is degraded under mostly anaerobic conditions. And when you degrade PHB or most other forms of carbon under anaerobic conditions, you're not making CO2, you're actually making methane. And so the true emission is really methane. And so again, think of carbon flux. CO2 comes in here, goes through this whole thing here, and comes out as methane. So, and again, you don't think about this when you're working in the lab. But when you're thinking large-scale commercial production, what this does is it pulls CO2 out of the air and it emits methane, all right? Which, as all of you know, is a gas that currently most countries are trying to reduce, not increase, all right? So that's one thing. The next thing is that when we look at the amount of plastic we use every year, about 20 million tons, and you then say, okay, great. If we had to make 20 million tons to service the current demand, how much land would that require? Well, turns out the 75 million acres we already have, we would need an additional 25 million acres of corn farming land. And that is the equivalent of New Hampshire, where I live, Vermont, Massachusetts, Rhode Island, Connecticut, Pennsylvania. Anyway, the combined area, not their farming area, wall-to-wall corn farming to produce enough sugar to make the annual demand for making plastic. So I had at that point left industry and joined the faculty at Dartmouth, a group that was working in the area of bioenergy. And they were mostly focused on ethanol and the whole concept of biorefinery. I'm sure you've heard all of this before. I was going to be the guy that does the materials, they do the fuels. So I show up there, I publish a few papers on this topic, and you can imagine I was not a particularly popular guy. So as a sort of career move, please do not view this as a blueprint for a successful academic career. In fact, the more media coverage this thing got, the more uncomfortable, shall I say, was my relationship to my immediate colleagues. And it sort of reminds me of this joke that you may have heard that supposedly happened during the French Revolution, where revolutionaries rallied up three royalists, a doctor, a lawyer, and a scientist. And they asked the doctor, as they put him on the guillotine, you want to go head up or head down? I don't want to see anything. Your head down. So they put him in. They release the guillotine, and it jams. They say, force majeure. You can go. Okay. Next comes what was the first one? The lawyer or the doctor? Doctor. Okay. Next is the lawyer. Lawyer also doesn't want to know anything. Face down. They put him in. It jams again. They say force majeure. Finally, the scientist is asked to whether they want to go face up or face down. And I've committed my life to understanding how everything works. I want to face what's coming. I'm going face up. They slide him under the guillotine. He looks up and he says, oh, I see what the problem is. So this is the situation I managed myself into. So there was a lot of interest in the area, and I think it sensitized people to think about this a little bit differently, and what may appear to be green may not be as green. But I was fortunate enough to advance out of that state and find myself at a time, or maybe I should say this a little differently. I'd spent 7 years of my life working in this area and had not even asked the question, what are the larger repercussions? Let's say we'd really succeeded in doing this, what would the repercussions be? So when you work on something for that long, and you then sort of have, at least in my opinion, very little to show for it, you are highly sensitized to the quality of the problem you're trying to solve. So while it was a waste of time in some ways, in another way it was a real wake-up call that before you spend any time working on a problem, really understand the quality of the problem. And we scientists have a tendency, we love solving problems. And so in a way we're like greyhounds, right? If it's fuzzy and it moves, it has to be a rabbit. Well, not so, right? And if it's a problem that is thrown at you and I want to solve it, you know what? All problems aren't equal. And so this experience acutely tuned me into the question of what's a quality problem. And by the way, sort of, you publish these papers, there's a lot of media attention, but mostly because it's a story like, man bites dog. Unexpected outcome, and the media, typically not very sophisticated, when there's something that is unexpected, they want to write about it. Fair enough. But that's not a productive life as an academic, to just poo-pooing what other people are doing, right? So you better go on and do something useful. So sure enough, the year was the year 2000. And as you all remember, this was just before we had the first version of the human genome being published. And there was a technology bubble that emerged at the same time that many of you will still remember. Most of you were like, 12 years old? in any event, it was a very exciting time. So exciting that companies like Celera that did something really unusual. They took human DNA and they sequenced it. Whoop-de-doo, right? They did just at a very large industrial scale what was an established technique. They were able to basically shotgun clone human DNA fragments, sequence them, and assemble them. In fact, assemble them based on a blueprint that was provided by a government effort. And you know there's a whole political discussion between free marketeers and how they want to see the situation. But in any event, what happened was human genome was being sequenced. Celera had a market capitalization of $18 billion with no product. In fact, not even a plan for a product. And the beauty is, in the hindsight that we have now, they still don't have a product. Okay? But that company was worth $18 billion in the first quarter of 2000. And the reason for that was that people very quickly understood that if we have the sequence for every single human protein, that is going to have some, and most people thought, a positive impact on the discovery of new drugs. And so now you can ask yourself the question, well, how would that play out? Well, it can play out in two different ways. It can play out in a way where a new receptor is discovered that does something that causes inflammation. Once you have that receptor, you can now clone it, set up bioassays, interrogate that receptor with small molecules, screen huge chemical libraries, exactly what pharma is good at, to find something that inhibits that may at some point lead to a drug. And we all know that stuff takes decades. Okay? So, in other words, this knowledge could lead to the discovery of new small molecules. Alternatively, it was already known at the time that many human proteins by themselves are drugs. Insulin being an example, erythropoietin being an example, many others, as you know. And so, in that case, when you know there is an undersupply of a particular protein, factor VIII or whatever it may be, and I have the human gene, well, it's not that hard to figure out that there is a potential drug that could come out of that. So when you look at this world now between small molecule drug discovery, this thing is like vibrating. Does that mean anything? Is this like the audience voting that they don't like what they're hearing? Oh, it's working again. So small molecules, or what we call large molecules, which are protein-based therapy. And to make life a little bit more complicated, 70% of all human proteins are not just a strand of amino acids that fold in a particular way to give it function. 70% of all human proteins are glycosylated. So they have specific sugar structures attached to their surface, and in fact, those sugar structures have a meaningful impact, an important impact on the properties of the protein. And we can go through the list of that later, but just take my word for it now. They're important. Good. So here we have human genome being sequenced, thousands of genes being available, people thinking, wow, there are going to be lots of drugs. The world I just described to you, small molecule, yes, impact, but long-term, new recombinant proteins, everyone was excited about that. And so very quickly, people realized that this new information is going to lead to a need to make more of these types of drugs. And so you can have sophisticated modelers and investment bankers go through all this, and they'll tell you, if we look at the current rate of attrition of protein-based therapies in the clinic, in 4 years, we're going to totally run out of manufacturing capacity. So there was a huge interest at the time, and again, any sort of analyst report you read, squeezing manufacturing capacity, they were all telling each other the same story. Bottleneck in manufacturing, yada, yada, yada. Okay? So, back to the core issue. 70% of all human proteins are glycosylated and require glycosylation to be functional. So this is human erythropoietin. You take off the sugars, it is worthless as a drug. Okay? So it requires sugars to be biologically active. So that's one issue. So if you take those sugars off, the drug is not functional. So you need to express it in a system that now replicates or produces human-like glycosylation, otherwise the protein is useless. The next issue is, if you remove the sugars that come out of a traditional mammalian manufacturing process and you analyze them, what you will find is that the product is highly heterogeneous. These cells are super sensitive to even the most subtle media changes, pressure changes. It's very difficult to scale up from a small scale to a commercial scale while maintaining this particular composition of sugars on the surface. And this, again, may sound trivial, but when you're spending millions of dollars doing a phase 1, phase 2 trial, and then you want to produce something commercially, and the FDA says, gee, guys, this peak now is twice as high, this one's a little bit lower, new composition. Show me that that drug is safe. So it was a nightmare, okay? And moreover, as you have this heterogeneous population, glycobiologists at the time were figuring out, you know what? Some of these sugars make the protein a lot more active, whereas others make it a lot less active. So again, that's why the composition matters. And we had virtually no control over the composition, okay? So the good thing is, if this is the most active form, at least you have a reasonably active drug. But if you're a little less fortunate, and this is your most active form, you're kind of out of luck. And we had no good ways of purifying them. Essentially, we were at the mercy of the host that we chose, and we all settled on a true improvement method of manufacturing, which was Chinese hamster ovary cells. The vast majority of these drugs are produced in Chinese hamster ovary cells. Okay? Good. So here's the problem. People thought manufacturing was going to be a big issue, and we need to find better ways of doing it. So there were two options. People were building large plants and spending hundreds of millions of dollars doing that. Another group of people, mostly animal geneticists, went and started engineering every single poor animal farm animal they could get their hands on. So you probably remember transgenic goats, transgenic chickens, transgenic rabbits, transgenic boar semen. Wouldn't want to have that on my resume, being the head of downstream processing at the boar semen facility. But in any event, if there was an animal, people, shrimp, so people were engineering animals to make recombinant protein. Okay? At the same time, people were engineering plants and started to think about how to make tobacco, various other plants, corn, to have a more human-like glycosylation. So I thought, people are already using yeast, in particular in the industrial enzyme business, where fungi and yeast are sort of the proven workhorses, where what is important? What's important is high yield, low cost. So if I were to choose a manufacturing platform, I would use something, A, that is compatible with current manufacturing methods, which as clean as you may be able to keep a goat farm. If you ever go to a GMP manufacturing plant and you go to a clean goat farm, you will see there are different sanitary standards, let's say, okay, that are not easily to consolidate in a fairly regulated environment. Good. So we thought, let's take yeast and use yeast to make recombinant proteins. But the problem, of course, is that yeast is an organism that is not able to make human glycoproteins. So in other words, the sugars are different. Not only are they different, the innate human immune system has evolved to have a lot of mannose receptors, because normally when we, as humans, see proteins that are highly monosylated, we have a yeast infection. And we want to get rid of that stuff as soon as possible. So yes, you can make metric tons of erythropoietin, but it will have a half-life of literally minutes because these mannose receptors will remove it from the bloodstream. In other words, you're never going to make a drug based on yeast-like glycosylation. So I, as an academic at the time, getting out of the biopolymer field, wrote these elaborate grants to NIH, the Whitaker Foundation for Bioengineering, NSF, the usual stuff, right? And they all said, listen up. This is a really, really complicated problem, and if anyone's going to solve it, it's not you. Because you are neither a glycobiologist, which is true, nor are you a yeast geneticist. It didn't matter how coherent the plan were, how it addressed all the other issues that people had encountered in the past. It doesn't matter. Our funding agencies are not very interested, and this is something to keep in mind, in allowing people to change their careers. It's normally, okay, you did your undergrad work in this area, then you do your PhD in a similar area, then you do your postdoc in a similar area. I can tell you, whenever I moved from one stage to the next, I almost insisted that I do something different. And it really gives you a much broader perspective. So if you have the opportunity of leveraging your expertise, but try to leverage into something where you learn something really new. In any event, wrote grants, they were all turned down, always the same story. So by luck, I was teaching with a colleague of mine who was the dean of the engineering school at Dartmouth at the time. And I told him about this idea, and it was a really cool thing. And it turns out that Dartmouth has a pretty good connection to a lot of people in the both venture industry and in the biotech industry. And upon having conversations with these people, they said, well, this is a really important problem. It's very hard to solve, but if you solve it, it's going to be really valuable. And this is sort of one message that I would want you to consider as you're thinking about your careers. The quality of the problem matters. In this case, because it was perceived at the time as being an important problem, you could also attract the capital to do very, very risky things. I'll get back to that a little bit later. So anyway, long story short, Hutch and I co-founded a company called Glycoproteins, raised a little bit of money, and within a relatively short period of time, by very methodically approaching the problems that others had encountered and understanding them, we were able to make progress very rapidly. So again, just to briefly sort of lay out what we did. If you take a gene of interest, shown as this little yellow sausage here, and you introduce it into the nucleus of either a mammalian cell or a yeast, what ends up happening is the protein is expressed in the endoplasmic reticulum, or it's produced synthesized translated in the endoplasmic reticulum. It is then shuttled through the secretory pathway to these COP2 vesicles into the early Golgi, medial Golgi, late Golgi, trans-Golgi network, and then finally secreted. The composition of the sugars on the surface here are a direct function of the enzymes the protein encounters as it works its way through the secretory pathway. Okay? So, all sounds pretty simple. So let's focus on what happens in the endoplasmic reticulum in yeast versus what happens in humans. Because if they're the same, there's no work to be done, which is great, right? And we can move on to the next compartment. So we're going to look at now what happens to protein synthesis and early glycosylation in the endoplasmic reticulum. Humans on the left side, yeast on the right side. And from now on, this little red helix, this rather ugly red helix here, represents a protein that has a specific sequon that leads to end glycosylation. Okay? So the yeast will, at the luminal side, the inside of the ER, synthesize this very specific structure of sugars, and each square here is a mannose, each hexagon is a glucose. You don't have to know all this, and the different coloring just tells you that there are different linkages. But we synthesize this structure when we recognize the sequon. There's an enzyme complex that then transfers this to the protein. So now you have a nascent glycoprotein. Okay? And as soon as it transfers, there are two enzymes called glucosidase 1 and 2 that remove these terminal glucoses here. And then there's an enzyme sitting in the ER called alpha-1 mannose that removes this specific mannose here. All right? So that's what we humans do in the ER. Synthesize protein, transfer the core oligosaccharide, trim it down a little bit, on it goes to the Golgi. Okay? We now look at what happens in yeast. And by the way, we did none of this work. This is us reading the literature and understanding this, but this is the investment we as a society make in basic research. Right? Nobody knew that this is going to have some potentially commercial utility. This is us spending a piddly amount of 0.3% of our GDP on figuring out how the world works. Okay? But that's the knowledge that we were able to benefit from. So when you look at yeast, you find that the yeast that brews wine, the fungus growing on your shower curtain or on your jam, and what happens in humans, early glycosylation is highly conserved. The same. So there's nothing we needed to do. But, as you will see in a second, once the protein is now shuttled and moved on to the early Golgi, those two pathways diverge distinctly. So let's go and look at, we've basically exited this here. We're now moving into this area here. Now let's look at what happens to the glycosylation in the early Golgi. Okay? So, protein shows up in the early Golgi as the structure we call man-8. Man-8 because it has 8 mannoses. If you count the squares here, it's 8 squares. And once it shows up in the early Golgi, there's a bunch of enzymes called alpha-1 mannosidases that then trim down all the terminal 1,2 mannose to give us what we call man-5. Once this mannose here is exposed, there's an enzyme called glutamate transferase 1 that puts on a glupnac. Once that is on, there's an enzyme called mannosides 2 that removes these two guys here. And without boring you with all the details, there is a very structured sequential set of reactions that are separated in time and space that lead ultimately to molecules that are terminally sialylated. They have sialic acid at their terminus. And when you have that, it gives you the ultimate half-life in humans because it not only increases the size to prevent renal clearance, it also charges the molecule negatively to, again, prevent or decrease the rate of clearance. All right? So that's what we humans do. So let's go over to the yeast and look at what the yeast does. So it receives this mannose structure from the ER, like humans do. But instead of taking mannoses off, what yeast will do is the exact opposite. It will start adding mannose. In fact, once it adds this first 1,6 mannose, all hell breaks loose. And there are manosyl transferases throughout the secretory pathway that in a rather indiscriminate fashion slap on mannose, creating structures in cerevisiae and baker's yeast that can have 100 to 200 mannoses, Christmas trees full of mannose. And in Pichia, they're a little bit smaller, but most yeast will do something like this. Okay? To top matters off and make them even worse, there's often phosphomannose involved, 1,3 mannose. Some of these are fairly immunogenic, in fact. So we can make boatloads of this stuff, but as soon as you have this attached to it, it's worthless. Okay? So I think you get where this is going, right? How do we make this look like this? And so our goal was to humanize the yeast, and so we went to the literature, and people had tried this before. As all our grant reviewers had pointed out, painfully, not only do you suck, but other people that are a lot smarter than you tried, okay? Tried for many years. So what they tried is the following. They realized that this enzyme is really important. This is a 1,6-mannose-transferase. It's encoded by a gene called OCH1. And a group in Japan under Professor Jigami in the early 90s managed to knock out this gene here. And when you do that, when you knock out OCH1, you essentially end up in a situation where this whole sidearm here is removed. I would characterize that as progress. Next, there's an enzyme called or a gene called MNN1 that encodes a 1,3-mannose-transferase. You knock that thing out, you get rid of this. Okay, so at least with 2 knockouts, we can get to a starting point where it's at least not messing things up. It arrests further development at a stage that is identical to what comes out of the human ER. So I would say that's good news. Okay? So next step, we need to make this look like that. How do we do that? Well, we clone in the enzyme that does that. It's an alpha-1,2 mannosidase. And the same group under Jigami that in the early 90s knocked out these 2 genes, OCH1 and MNN1, then decided, let's clone in a mannosidase and trim this to make it look like this. And this is where the story gets really interesting. So they found an alpha-1,2 mannosidase, cloned it in the hope they would get this. And then I would say in a landmark paper in 1998, Chiba et al. Described the following situation. When you take a baker's yeast that has knocked out OCH1 and MNN1 and you clone in this mannosidase from this fungus Aspergillus cytoi, what you end up getting is, top line here is man 8. Remember, that's our starting material. And we want to get to man 5. So they have a lot of residual man 8, man 7, man 6, and a little bit of man 5. So in other words, they were able to partially get the processing they wanted. And you read the papers, and they're like, we break the cells open, we do membrane preps, we assay for the enzyme, it's there. We do immunocytochemistry, we have antibodies against it, we show it's in the yeast and it's in the right place. Okay? But whenever they do it and whenever they express the protein, they can never get beyond like 25%. So this is one of those moments that you don't have very often in your scientific career where 5 years in the lab can save you an afternoon in the library, which in this particular case played out as follows. So we knew of all this work. We knew that they sort of got to the starting point, and we knew that, and you read the papers. They were like, maybe we don't have enough enzyme. They already were using a yeast 2-micron plasmid. They had hundreds of copies, strong promoters. There was no question that they were making this stuff, but it wasn't doing anything. So whenever you see a situation where you're making it, but it's not active, you have to ask yourself, well, what environment does this enzyme find itself in? And so I went back to the library and looked at this enzyme from Aspergillus cytoi. And I looked at a very simple table. It was a paper published by Yamashita in 1980. It was almost 20 years before I started working in this area. That did a simple experiment and looked at the pH optimum of this enzyme. And what you can see is that at pH 7, this thing is essentially worthless. It doesn't do anything. Okay? And why? Because fungi secrete mannosidases, in an environment that they typically acidify. Okay. But when you then take that enzyme and put it into a yeast where the pH in the ER and Golgi, if you look at the pH optimum of most of those enzymes, it's around 6.5, 7. So presumably nature evolved the enzymes around the pH that they find themselves in. And so this was completely out of its element. You can express this all day long. The fact that you're running a reaction at pH 7 essentially rendered it inactive. So we then said, okay, you can't try this one by one. There's a certain reaction we want. It's a 1,2 mannositis reaction. Do we know which alpha-1,2 mannositis, of which there were many known, is going to work best in the yeast that we were engineering? We had no idea. And that's what engineers are good at, right? They say, that's the problem. What do we know? What do we not know in this case? There was more that we didn't know. But if you don't know, at least do an experiment where you try different things. So what we said, and this was heydays for undergrads in my lab, because anyone that had like any semi-useful pair of hands could walk in the door, here's the paper for the mannositis you're going to clone. So they designed their first PCR primers and all this stuff, and off they went, and big success. We were able to build libraries of mannositis in very short order. Okay? Step one. The next thing that people didn't know is, if I take a human mannositis and I clone it into yeast, there must be some signaling involved. In other words, the protein sequence must encode localization. Do we know what we know for the genetic code, where codons in yeast are the same codons that we find in humans? For signaling to certain parts of the ER was unknown. So what we did is we took all the signaling sequences out, built large yeast libraries of those, and made combinatorial libraries of all these enzymes with all these signaling sequences, and we had no idea what worked. But we at least did the experiment not once, but 1,000 times. And guess what? When you start looking for productive combinations of mannositis and targeting sequences, you find some that essentially process to complete fidelity. Okay? So having set up the system now of knowing what we know and knowing what we don't know, asking the question at a broad enough scale, developing the analytical tools, this by the way is mass spec, which is much, much faster than the HPLC analysis I showed you before. So again, you have to set all the pieces up, but once you have that, you can plow through it pretty quickly. And so in our case, we ended up, over the course of 6 years, engineering a yeast that was able to make this at essentially 100%. And then we got to the last step of sialylation, where the problem is yeast doesn't even make sialic acid. So you have to engineer in the pathway to make sialic acid. This yeast had 5 gene knockouts, 9 genes cloned in to make this, and then we still had to do this final step. So we had to engineer in the pathway to make sialic acid. Oh, more bad news. You can only make that in the cytosol. You still have to get it into the Golgi. So you have to clone in a transporter. Again, that was not trivial to get it inside. And then you have to optimize around the sialyl transferase that you, again, localize properly to get sialic acid transferred to give you this. Okay? So you need this as a precursor. You need to make CNP sialic acid. You need a transporter, and you need a transferase. And they all need to work very efficiently to make that happen. So if this is pick your wild type, again, you see a heterogeneous mess of manos. By 2005, we were able to make strains that make exquisitely pure glycoforms on a protein. And so this yeast, you put any gene in that has a glycosylation site, it will only put on one specific glycoform. All right? And so by 2006, we were able to show that you can make predominantly sialylated species in yeast. And when you treat this with sialidase, which cleaves off sialic acid, you can see it collapses back to the precursor, so we knew we did the right thing. Okay? So at that point, we had humanized yeast, which everyone said was impossible. And we had a vehicle, and it's a lot easier to clone in yeast. It's cheaper and all this stuff. One thing that I'd also say, being in the making stuff cheaper business, in my experience, is not a good business. It doesn't work as well as you may think it will. And so the proposition here, oh, CHO is so expensive, and we're going to make your stuff for cheaper, that did not go over that well. So what did, however, go over very well is something I told you before, that when you make stuff in mammalian cells, you get this heterogeneous stuff. And we were able to say, oh, wonder what this does. Make that protein with only that sugar on it. Wonder what this does. We could make each individual glycoform, and then you could put them into animals or into appropriate assays and figure out which one is the most potent. And that's what the action was, to show that you can make molecules that are exceedingly more potent than what you get out of this. That's where the value was, and that's what ultimately led to a situation where several of our partners wanted to acquire us, and so we published, obviously, much of this, and then by 2006, Merck decided to acquire the company. And so I was out of a job again. Great. So, and they took it, and it's a longer story that we can talk about another time, but in any event, that was 6 years of my life, starting with something completely new, yeast for that matter, and carrying out this work, ultimately leading to there are 2 drugs in the clinic now based on this technology that you could not make any other way. And so we'll see how they work, but it was a pretty good story. So what happened? What happened here is that we as a society make an investment in basic discovery science. As I mentioned before, 0.3% of our GDP goes into the funding agencies, the NIH, NSF, and more now private philanthropic organizations like the Howard Hughes Medical Institute. Put it all together, $40 billion. Okay, 0.3% GDP. Okay, this is about 1/10 of Medicare fraud. Just so you know, this is like when everyone gets free strollers and stuff. That's what we're wasting 10 times more money on than figuring out how the world works. So there's that piece. And most of this money here is really dedicated to figuring out how things work, not necessarily meeting a human need or anything of that sort in terms of like coming up with a cure, right? But there are human needs. And in fact, basic science very closely ties to human needs at many different levels, not just curiosity or, why are dinosaurs so close to chickens and that kind of stuff, which is interesting, but it doesn't really solve an immediate human need. So, but there's a whole other funding mechanism that in fact is focused on turning this into something that helps human needs. And as always, you need capital to do that. And in fact, there's a fair amount of capital available to do that. So there are certain foundations like the Bill and Melinda Gates Foundation. They don't care about basic science. They identify needs, and then they go out and find the basic science that may be needed to come up with a cure or a treatment for something that we currently don't have any solutions for. The other thing you need a lot of, VCs in the U.S., life science focused investments are about $6 billion out of $20 billion total. The other 14 are more for Facebook kind of stuff, social media, clean tech a little bit, less and less. But anyway, $6 billion come from venture, and then a whopping $65 billion that pharma spends on turning this into drugs that help humans. So again, $40 billion, all the universities in this country, all the teaching hospitals, that's this. And the amount of capital that is deployed to move this over here is actually significantly higher. You need a lot of engineering. And in fact, engineers are the people that understand this, and then are able to come up with something that really solves a meaningful problem. I can tell you, the older I get, the more I'm sensitized to this particular issue. And then there's this elusive concept of innovation that, as you know, everyone talks about, right? And everyone has a different definition of. I personally like this definition here by Peter Drucker, who's a very well-known management and behavioral scientist, who said, innovation is a specific instrument of entrepreneurship, the act that endows resources with a new capacity to create wealth. So what you see on the left here is silica or sand. What you see over here is a CPU, a microchip that has huge computational power, but it's not the raw material in here that has any value almost. In fact, it's worthless. Right? It's really the innovation that is creating something that has properties that are entirely different than what you see on the left here. Or another example is this here, where you can take all these raw materials and you can make something that some of us enjoy, but there's a lot of engineering and innovation that goes into turning this into this. The example I gave you before, right? Take baker's yeast or a yeast, and you teach that yeast to do something differently. Can't see the difference really. But Merck bought one of these two for $400 million. Okay? So, again, the way I would describe it now, as bioengineers, we live in this world, and we don't live just in this world. I mean, we live in a world where there are human needs, and we need to be able to figure out how to connect these two. And the sad reality is there are really good ideas that can't find the capital to do something that has impact. And so we are very focused on technical problems, but many things are not happening not just because of technical problems, because they can't be financed. And so one has to develop a much more holistic view, in my opinion, if you want to have an impact over here, not just about understanding the science, but what can be financed, what's the engineering piece that needs to come together to convert some of this to the stuff over here, and then surely innovation, not just in how to implement the science, how to make the technology work, but also how to finance it, business models. There's a lot of innovation in business models where you can do things today that weren't possible maybe 5, 10 years ago. So we're getting better at all of these things. And so I think it's a very exciting time to live in and to start a career. So okay, finally I made it to Central Europe through the Fertile Crescent. And I'm going to now just tell you one last story before we wrap it up. So after Glycoproteins was acquired by Merck, I had to agree to be a consultant for a year, which was fine. But again, you learn a lot. You learn a lot how pharma thinks, what they do, how they do it, what they're good at, what they're not good at. At the time, the emerging picture was, again around 2007, is the future is biologics, protein-based therapies. Because the small molecule tree has been shaken very hard, and it was harder and harder to get good drugs that are specific, even very successful drugs like Vioxx. Guess what? Once you deploy that over a population of millions and millions of patients, there are going to be some people that have an adverse effect, and that can kill otherwise very efficacious drugs. So, in any event, pharma wanted to be in biologics. I saw it from within Merck, how they were thinking of getting access to antibody discovery. Well, first of all, when you think about biologics, there is the bucket of recombinant replacement proteins, erythropoietin, glucocerebrosidase, insulin for that matter. That's one bucket. And the other big bucket is human monoclonal antibodies, where essentially we now have the ability of targeting any human protein that's on the surface or in solution in a very specific way with molecules that have long half-life, high degree of safety. So they are a really awesome modality to modulate biology and ultimately come up with therapies. So, in recognition of this, Dane Wittrup, a colleague of mine at MIT, Eric Anderson, a former student of mine, and myself, we started an antibody discovery company that I'll just tell you a little bit about. Again, not to talk about the science, but how you can do different things in your career, provided that you find problems that are relevant. So if you look at total sales of antibody-based therapies this year, 2014, $60 billion. I mean, that is a massive economic impact that this single drug class has had both on human health but also commercially. 4 of the top 6 drugs this year will be antibody-based. And so the good news is there's a high degree of validation in the clinic, high degree of validation in the market. And so what happens now is people figure out, hey, this receptor may play a role in oncology. What happens? Every drug company that is oncology wants to develop antibodies against this target. So the competition is really high. And that's typically also something that people shy away from. I mean, this oncology target here, IGF-1R, in 2009, there were 6 programs. We now know of 11 programs just targeting that one receptor. So 11 different pharma companies are trying to develop an antibody that hits that target. Okay? Huge competition. So we said the way we discover these drugs now, either by taking animals, injecting them with antigens, waiting for, I mean, some of you may have raised monoclonal antibodies. It's a not so pretty picture. It takes a long time, uses animals that may not give you what you're looking for. So it's very cumbersome. So we said that's the problem we want to solve. We want to have a better way of getting from here's a target to here is a monoclonal antibody that hits that target with high affinity and has all the properties that you're looking for in a drug. So what we did is we took yeast, both because Dane, my co-founder, and I had a lot of experience in expressing proteins in yeast, and we had to heavily engineer yeast to actually make high-quality protein. As we discussed before, there's a lot of O-glycosylation going on that then leads to misfolding. So if you make antibodies in wild-type yeast, they come out in a schmear top to bottom if you run them on a gel, right? Some assembly required, but actually quite a bit of assembly required. So you don't get high-quality protein. So you have to engineer the yeast to to allow you to do that. And then we built expansive libraries of human immune repertoire. So each one of us sitting in this room has roughly 10 to the 8th, so 100 million different antibodies that are produced at any given time. Of course, in response to an infection, that changes, but that's the normal immune diversity, 10 to the 8th. And so what we did is we synthesized diversity that is about 2 orders of magnitude larger, so a little bit in excess of 10 to the 10th. In other words, we took the immune system or the immune diversity of 100 people and then put them into a format where we were able to express them in yeast, where each yeast cell on the surface now expresses a fully human IgG and presents it. And of course, we don't have just these 6 cells that you have here. We have literally 10 to the 10th. So in other words, 10 billion different yeast cells, each one of which expresses and presents on the surface a fully human IgG. All right, that's good news. So now if you want an antibody against something you care about, let's say an oncology target that is overexpressed on a particular form of breast cancer, you make that antigen as a protein, you fluorescently label it, you throw it into this library, and those yeast out of the 10 billion that by chance make an antibody that bind to this thing are going to sequester it on the surface. Because you've labeled the antigen with a fluorescent label, it's going to look like this. So now you have a separation problem. You have 10 to the 10th yeast, some of which are going to look like this, and you want to get those out at the expense of the others. And as you know, there's very powerful instrumentation known as a fluorescent-assisted cell sorter, where you can take a population of mostly red cells and fish out the few fluorescent ones by running them single file through a capillary, interrogating them with a laser, and finding the ones that fluoresce. And this can be done at 10,000 events a second. I can basically do 10,000 ELISAs in 1 second. Okay? So now you sort out the ones that are binders. These go in the trash. The binders are collected. And then we individualize them, grow them up, secrete the antibody now, no longer presented on the surface, and we have a fully human antibody against the target of interest. Okay? Completely changed the field of antibody drug discovery, where now we have a process of having a library of roughly 10 to the 10th, which I told you about before. These little boxes here are supposed to be yeast cells. These little things on the surface are supposed to be antibodies. Yes, this is not to scale, but just to illustrate that there are antibodies on the surface. You throw in an antigen that is fluorescently labeled. Those yeast that have antibodies that bind will now sequester on the surface. The whole thing goes into a fluorescent-assisted cell sorter that gives you an output that looks like this. A sea of non-binders, a few that fluoresce and bind. This goes in the trash. This gets plated out in 96-well plates. We grow it up. By changing the growth media, it no longer presents on the surface, but is now secreted into the medium. So here they secrete. This takes from here to here, takes 3 days. You then purify, and at the end you end up with a 96-well plate of fully human antibodies that bind to this target. So something that used to take months, if not years, is now a matter of 4 weeks from give me an antigen to give me a potential drug that you can then interrogate in your assays. Okay? So, yeah. All right. So it took us about 2.5 years to build this capability here in its sort of first prototype form that worked. And again, back to that point I made earlier, that if you are identifying a problem that people really care about, you, once upon developing a solution to that problem, should experience a lot of people coming to you and saying, hey, can you solve my problem for me? And that's exactly what we found. So we founded the company in 2007. 2.5 years later, we had our first partners. We started partnering with Merck and Roche. And then over the course of the next 3 years, we signed up half of top 20 pharma companies to come to us and say, can you please discover drugs for us? And so we did that and turned this from a venture-backed company into one of the very, very few profitable venture-backed companies. And at the same time, people realized that this is so much better, so much faster, that when often it's academics or entrepreneurs that have really interesting ideas around very important disease areas, they would come to us and say, can you work with us? At the beginning, we couldn't afford it because we were relying sort of on the revenue coming from these guys. But now that we're financially independent, we started more and more working with these guys. And it's just, you're tapping into a talent pool where people have fantastic, exciting ideas about disease biology, how it relates to targets, whether you can target them with an antibody. They are all coming to us now. And if they don't have the money, we in fact help them found companies where our venture investors are no longer able to put money into Adamant because we don't need their money. And so we're saying, well, why don't you invest in this company? So we founded a company in the infectious disease space that's based in Vienna, Austria. We just founded a company in the neurodegeneration and Alzheimer's space. Again, very interesting, very well genetically validated targets where there's a good hypothesis if you hit this thing with an antibody, you may get a therapeutic benefit out of it. So we founded a company called Elector, which is in Mission Bay here in San Francisco. And we're in discussion with a whole bunch of other folks. So with that, I'm going to wrap it up. And thank you for your attention and open the floor for any questions you may have.
Thank you very much for this very insightful talk. Thanks for sharing your experience with us. I'm sure all of us appreciate it a lot. We have time for one or two very quick questions because lunch will be cold otherwise. So if there are questions, please come up with them now. Mira?
Thanks for a really great talk. To hit on something in the earlier component, are the bio-based plastics still as unfavorable as they were in 2000 when you published those papers? I'm curious what the current state is.
So in life, answers to those types of questions require an agreement on accounting principles to start with, which in the financial world we kind of have, right? In this world we don't, because you have large financial interests of people that have already invested boatloads of money into this, that want to keep on telling the story how green this is. They have no interest in having a real, honest discussion about what the trade-offs are. They want to ride the pony of we're green and you should be paying more because it costs more to make the stuff, because you're being a careful steward of the environment. So the short answer is I don't believe so. There's no data to suggest that it is any better. And also in my analysis, I assumed like 100% thermodynamic efficiency at every step. So yes, you can improve things, but you're never going to go beyond the 100% thermodynamic efficiency. So I don't believe so. And I can tell you one more anecdote. The people at McDonald's, they many years ago had conducted a similar study when they were still packaging burgers in polystyrene clamshells. And I don't remember, they were polystyrene foam. They were yellow. And they did an analysis to ask the question, is it better to use polystyrene or cardboard? And the analysis came back, and the analysis clearly showed that polystyrene is favorable. But they said, look, ask 100 people on the street what is more favorable, and 99, or unless they run into me, maybe 100, right, will tell them that the cardboard naturally derived renewable packaging is more favorable, and they're not in saving the planet business. They're in the marketing, promoting themselves as being good to the environment business. And so a lot of these decisions, unfortunately, do not undergo really meaningful scrutiny in terms of understanding the real trade-offs, in my opinion.
Actually, Mira reminded me of something that I thought was pretty interesting that you said when attracting venture capital and sort of selling your idea. You said that basically marketing something as being much more efficient in terms of specificity was better than doing something cheaper, right? So making something cheaper, people didn't seem that interested. This is when you're talking about your yeast glycosylation.
Yes, so maybe I should be more careful. In the drug discovery area.
That's what I was going to ask. Do you think that same sort of mantra would apply to something like biofuels or like a renewable?
No, I mean, the real challenge is fuel, is the cost, right? But the cost, again, cost ultimately is tied to consumption of something. You're paying for something. And whether it's dilute acid treatment, guess what? You're going to have to buy sulfuric acid. And there's a certain amount of sulfuric acid you have to buy to make a certain amount of fuel. And when you add all those costs up, that's what ends up being the cost. So reducing costs, reduce reduces material input, and presumably makes it a better process. So there you're in a very different environment in the bioenergy or biofuel space. Just as a reference point also, for drugs, the final cost that manufacturing constitutes in final sales is like 5%, right? So if you improve that by 10%, nobody cares, right? On drugs that are selling for very large amounts of money. I mean, Seridase, the drug used to treat Gaucher's disease, sells for $350,000 per patient per year, right? Of which 5% is manufacturing, so nobody cares at that point. Right? So, yeah, I'm happy to have lukewarm food if you are.
Yeah.
I was wondering what the process of yeast was where you humanized the glycoprotein content.
It's a very good question. In fact, in diligence, when we were trying to raise money, people said, well, can you do all this and still have a viable yeast? What pointed in a direction that may be possible is people had done pretty serious deletions in early glycosylation enzymes and could show that you could monkey around with the stuff in a pretty severe way and get a fully viable yeast that doesn't have any problems. What really turned out is that, in fact, as we did all this engineering, the yeast was compromised and was not as healthy as a wild type, which then led us to go through a process where we take the yeast and we run successive fermentations at large scale, pick the ones that are robust to genome-wide sequencing, identify the mutations that matter, put them into the strain, and everything was hunky-dory. Lunch.